All right, distinguished participants, ladies and gentlemen, let's get to probably the last technical session for this conference, and it's going to be talking about technology tools for research. We're going to be looking at emerging technologies for research integrity. Technology tools for research, emerging technologies for research for research integrity, I beg your pardon. And our speaker is Dr. Kaylin Adipietro, who holds a PhD from Duke University in genetics and genomics with a certificate in cell and molecular biology. She entered the UMB research community in 2013 and joined the UMB Research Integrity Office in 2022, working under the UMB Research Integrity Officer, Dr. Stefan Viges, who just presented to us. Her responsibilities on campus foster research integrity at all levels of training. Dr. Adipietro participates in responsible conduct of research, RCR trainings for postdoctoral fellows and junior faculty, provides training in RCR, research misconduct, and rigor and reproducibility to various student academic programs, and leads departmental RCR presentations to senior UMB faculty. Her enthusiasm for good practices in scientific research has helped promote research integrity throughout the UMB system. In addition to promoting research integrity through discussion, Dr. Adipietro participates in the institutional process of addressing research misconduct when allegations arise. She works with the UMB Research Integrity Officer to conduct investigations with adherence to institutional and federal policies. Dr. Adipietro is a member of the US-based Association of Research Integrity Officers and attends monthly regional meetings, monthly special interest meetings, and yearly conferences to engage with other specialists in the research integrity field and to stay apprised of best strategies and practices for promoting ethical research. Distinguished participants, ladies and gentlemen, please let's welcome to the EXCEL-RITE conference, Dr. Kaylin Adipietro. Please give her a round of applause.
Thank you. Can you guys hear me okay?
Excellent. We can see your slides as well. Please take it away.
Okay, wonderful. Well, thank you for having me. It's a pleasure to finish off the day before sort of the final note and lecture keynote. So I will be following up and sort of supplementing what Stefan, Dr. Wieck had spoken about, and a couple of other topics that we've already hit upon. But I'm going to introduce sort of the technology behind what we do in the Research Integrity Office and give an overview of what types of tools are available for integrating into the entire process of accountability and compliance. So the technology tools that exist are for every level of scientific research. So from your institutional level, you're looking at data collection when you're in the laboratory and what you do with that data in terms of managing and sharing. There are online training modules that exist to support responsible conduct of research training and different types of training for compliance-related topics, as well as auditing and monitoring systems that are used within the OAC office in general. And when you go to the research integrity office level, you're looking at a different set of technology tools, primarily looking for a system that will manage cases. So there's a uniform place to input anything related to a particular case for people to access. You're looking at tools that are necessary for gathering the data and the email, so the sequestration process that Stefan spoke of. And then things that we do on a regular basis involve forensic software, image manipulation detection, detection of plagiarism, and also the hot topic of artificial intelligence and how it relates to what we do in the office and really at the university. So we'll start just briefly going over the institutional level. Since a lot of people have touched on these topics, it won't be as involved, but starting with data collection technology. So I was a scientist prior to this position, and I am used to looking at a laboratory notebook. So this is an example of a laboratory notebook from the 1860s and a sort of more modernized laboratory notebook at this point. But the existence of an electronic laboratory notebook is something that a lot of universities are pushing for at this point. And what that does essentially is allows lab members to replicate what they would in a paper notebook. So you're spending the time to enter your protocols, your observations from a particular experiment, and taking down notes, getting the data in there, all of those types of things that you would normally be doing with a pen and pencil. I actually still prefer the pen and pencil method. We did not transition to electronic laboratory notebooks as a requirement at our university, but a lot of places have either begun to require it or have at least offered the option to their faculty. What is an electronic laboratory notebook good for? Essentially, data security is a component of that. So when your laboratory floods from the ceiling and ruins all of your paper notebooks, the idea is that you should have something backed up on a server somewhere or an external drive, but those laboratory notebooks might be ruined and you might lose all of your data. And so a very simple definition of data security then, not something that's getting shared with people that shouldn't have it, but just keeping your data secure from a disaster, very simplistic. The electronic notebook also supports the auditing process. So if everything is centralized in one sort of program, it will be easier for our office or for a compliance office to come in to examine things and make sure that the process is being followed. This also promotes collaboration within the lab and outside of the lab, allowing people really to share and importantly to search within data. So if you're looking for someone's experiment from 3 years ago, you can actually search by date or search by a keyword term and find that information as opposed to going back through a physical notebook to locate something. And the electronic notebooks will manage inventory and supplies. It's pretty useful in terms of lab management. But the things to think about with electronic notebooks: what type of science are you doing? What do you study? Are there institutional policies that are associated with using an electronic laboratory notebook? The security level of your data, is it in clinical trials? Does the data need to be de-identified? Is it something that really should be in a program somewhere? And then, of course, most of these things cost some version of money. So a budget is going to be, or cost is really going to be a limitation of a lot of the things that exist on the technology aspect of things. So if anybody's actually interested, Harvard has sort of this matrix that they've put together. This is just a close-up of it. Essentially all of the available electronic laboratory notebooks and sort of what it is that they offer. So there's a lot of information out there about ELNs. I don't recommend or not recommend using them. It's really a preference for people working in the lab and what type of time and effort gets put into training on these. But just for example, sort of an up-close lab archives is something that a lot of universities use. So you can create a notebook that lists your cell culture, your crystallography, and things about raw data files. And what's nice about these programs is that they do integrate with commonly used software, GraphPad Prism, your whole Microsoft suite of things, PubMed, which is the search engine for literature here, PubChem for chemical drawings, etc. So it can be very useful, but there is a learning curve there. And again, it's not necessary as long as someone is trained to generate their data on paper and have a good way for keeping track of that information. And it's not necessarily needed. We heard a little bit yesterday and today about data management and data sharing. And the idea of sharing data sort of outside of the university and inside promotes scientific inquiry, but also scientific integrity. So if someone is able to view your raw data source, you're more likely to keep it honest and have people be able to look at the data and assess it themselves in case there's a mistake or honest error there. It encourages new research to happen, that collaborative process, and it really protects, I guess, the data, the university, and the person from that idea of there being faulty data. So data storage and management, there's a lot of places that data go from start to finish in a publication, and that can be hard to keep track of. So at the university level, at UMB, we're using a Microsoft-based application. We have access to something called OneDrive, which everyone should have access to if they have an Outlook or a Microsoft account. And then SharePoint is a sort of a Microsoft program that allows you to create folders for sharing information with specific people. Other types of servers sort of on the cloud that people like to use in lab are commonly known: Dropbox, Box.com, your Google Drive. And then again, if you're using an electronic laboratory notebook, there's usually a cloud program associated specifically with that. And something else to think about with data repositories, then, this is sort of the broad sharing of data. So the NIH in the United States had implemented a data management and sharing policy that more broadly required scientists to share the data that are being used for publications. And so we are now required to put that pertinent data into repositories. And so things to think about are there discipline-specific repositories for data? Is it something general that can be added? Do you want to keep this within your institution? And whether or not access is open or controlled. And these are just some examples of the programs that exist out there. Some of them are free, some of them you pay for, but again, it just depends on what type of question, I guess, you want to ask and how you want to store things. Something else we heard about yesterday, and a different type of technology at the institutional level, is the idea of promoting research integrity in these online training modules. These are extremely helpful because they are widely available. They can be tailored to a particular university. So CITI training, the Collaborative Institutional Training Initiative, this is something that was spoken about yesterday. This is the Center for Bioethics Research in Nigeria, and this website, and just the talk of being able to take these courses, having your RCR courses and those particular modules available online is a great place to start in terms of training. So having online training for research integrity and responsible conduct of research is greatly important, but the idea of supporting that with in-person trainings and coursework, as well as ensuring mentorship, is what it should be. And I know that's difficult, but just making sure that an entire research environment is conducive to people learning and not making the assumptions that everybody knows what they're supposed to be doing. That's how we can further train people in research integrity. So online training modules are useful. We come into compliance auditing and monitoring systems. So within our university, we are again using an external system. This is called Cicero. What this is responsible for, essentially, it's a system where you can upload your protocols for human and animal research. This includes institutional biosafety committees also, so any sort of cell culture work that gets done. General clinical research, I believe, also is included in here. Radiation safety and conflict of interest. And so you've got one location for uploading protocols for review by the committees and boards whose job it is to make sure that you are maintaining scientific standards and ethical conduct. I know that the IHB Nigeria and the IRCE. I apologize if I'm mixing up all of my letters. They have an ethics board that reviews protocols and things like that. And I'm unfortunately not sure what system is in place, if there is one, to upload protocols or how it is tackled there. But this is a good way to centralize that information and to keep track of things from one step to another. So looking now at the actual Research Integrity Office. So we're a small office on campus, even though UMB is a large institution. So Stefan and I are responsible for the School of Medicine, the School of Pharmacy, School of Nursing, School of Dentistry, social work, all of the campus schools that are a part of the UMB system. So it is just the two of us. Other institutions, he mentioned Duke, which is my graduate alma mater, has an office of approximately 20 people at this point. And so by institutions' sizes, you can scale up or down, but I think we do a fairly great job just being two people at our institution, which is very large. But we do have a lot of things to manage. And so when you're talking about the Research Integrity Office, you're talking about managing cases, the email and data sequestration, the forensic software that's needed to look at the data that gets sequestered, and what we do with the data, which involves image manipulation detection, plagiarism detection, and the use of AI. So you saw this slide a moment ago, which is just outlining our process, which involves different technology at different places. So your preliminary review, you're looking at an allegation that someone has submitted, either by calling you on the phone, sending you an email, or going through our automated system, which will be my next slide. And then you're sequestering evidence, going through that evidence, and trying to find data that are related to an allegation, and whether or not that be source data that can show this was an error, or source data that doesn't exist, or data that shows somebody has manipulated something. So for our case management system, we use something called Ethics Point. This is called the Incident Management Program. And what this does, it acts as our hotline. So if you don't want to send an email directly to the OAC Research Integrity Office or to Stefan or myself, you don't want to be known, you can go onto this hotline. So Stefan had mentioned that this is basically at the bottom of every single web page related to UMB as a university. You can go in and anonymously complain about something. It should be hopefully specific. So I noticed that the person I'm working with has decided to falsify an image or fabricate some data. Here is the publication, or if there isn't a publication yet and this is happening in lab, it's something to bring our attention to, and we can speak about it before it turns into an actual case. But this is sort of what the interface looks like. So open a new case. It allows you to input a bunch of information about, where a complaint came from, what information we have about it, what is that person's relationship to the university. This is currently what our caseload looks like, although this might have changed. So this is the number of closed cases versus what's in progress and what is sort of on hold. And we go through, this allows us to have a source of metrics for output and how many cases we have open, how long it takes us to open and close a case. And Stefan was speaking about how that can vary from a fairly short time frame to something that unfortunately lasts much longer than what is suggested or recommended within the guidelines and sort of federal policy and university policy. But at that point, the purpose of these investigations is to do a good job figuring it out. So if we need to take more time to do it correctly, we are allowed extensions. So A 120-day time frame for an investigation can sometimes turn into 2 years, and that's a long time, but as long as we're doing things properly, that's really sort of the main point there. Again, within this interface, then you see that ethics point, hotline and non-hotline. So our source of the allegation, it assigns a case number, a date that things were opened, and these are all research related, whether or not they're in progress or closed. So these are all things that we can keep track of and we can share with the appropriate people within the OAC office, so people that need to know the information. So this is extremely helpful, both then as a hotline source and as a program that tracks cases all the way through the point of closure. So when we open a case, when we've made it through sort of this initial analysis and before we formally start an inquiry process, we're going to sequester the emails from a particular respondent or a person whom the complaint is made against, and we're going to sequester data. And it is recommended that any research integrity office that exists become very good friends with the IT department. So we work very closely with what is called the Center for Information Technology Services at our university, or CITS for short. These are the people responsible for maintaining our whole email system on campus, for maintaining lab servers and servers across the university. And they are significantly more knowledgeable about technology than I am. And so we usually have them, we can request to archive emails from a respondent, and those are normally provided to us as something called a PST file if you're using Microsoft Outlook. Additional information they can provide us with are the Microsoft OneDrive folders, which are sometimes used to store data. Any sort of university-run server that we have access to, they can provide us with access. And then we actually need them to come with us to remove hard drives from computers within the laboratory. And so whether or not that's somebody's personal laptop, somebody's work laptop, or a computer system that is hooked up to a microscope or a plate reader or some sort of computer dedicated to collecting data where something important might be. They will take those hard drives for us and make copies, which I will speak about. Physical sequestration also occurs. So if we've got a straight up regular laboratory notebook that has somebody's Western blot images or some trace papers taped into the pages, those, if it has data that pertain to what the allegation source is, we will take those notebooks as well. We keep those. If the respondents need copies of that information, we will provide them with a copy of that, but we maintain the regular data or the source data within our office in a secure way. So we are the only people with access to it at that point. So what do you do with all of this information? When we acquire the emails, you're going to want to search emails. So I have this sort of broken down as fairly basic ways of doing things and then more advanced ways of doing things with programs that may or may not cost money. So hopefully this will be helpful to someone in the future. If you are working from a Microsoft-based platform or any just sort of email platform, you can make copies. So if the IT department is giving you copies of these PST files or message files.msg, you can just open them in the program, in the email program itself. So you would have a copy of it and you will just essentially make the program open those documents for you. And you can save copies of those files as a PDF version or something that you will be able to use later. If you find an email, that says, I faked this data, then you can save that email as a PDF so you could go back to it later. Then there are technology programs that exist to sort of open these files for you. So Mail Examiner is one of these programs, and eDiscovery is something that, again, also works within the Microsoft system. And both of these you can use for email analysis. So you can go in and search your key terms. It will open an entire archived file of these emails, and you can search as if you are that person's email. So this is helpful for looking for attachments that might be containing data, or an email back and forth between people sharing data images, and whose excuse me, version of something that gets published, if that is trackable. So I mentioned we copy hard drives, or SITS does for us. So they will remove a hard drive from a computer and copy that information. There are a couple of different ways to do this if you do not have an IT department or the ability to use sort of more complicated software. In this case, you're going to copy the hard drive of a computer of interest onto an external drive. There are ways to be more careful about this, and if you have an IT department to be responsible for that, it's probably a little bit better. You can make 2 copies, so you will return the original hard drive back to the lab space. Your IT department will keep a copy, and then you will have a copy to work from in the Research Integrity Office. And you don't need to copy things like program files that help the computer run, but you do need to be able to find data and search for things that you might be interested in. If you want to get sort of more into making something called a forensic copy, that's where these other software come in. So the IT department will take the original hard drive and make an identical, basically just shot of what that hard drive is. And it's something called a forensic copy. So there are ways to ensure that the forensic copy is identical to what the hard drive is that you're copying. In theory, you can do that just regularly, but the forensic copy, and Stefan is probably going like this right now in my explanation, but it is a snapshot. It is exactly what was happening on that hard drive at that moment, all of the information that is there. And that's usually comes as something called an EO1 file or an image file. And these are the different types of software that can open that information for you. Once we have those hard drives copied, we need to look at the data. How do we do that? This is called the data triage sort of example. You can open up that hard drive that you just copied, and you can search it using your regular computer. And whether or not you will have the appropriate files to open those files that you found is possibly not. A lot of these files can be image files that were specifically saved as data to only be opened with a particular microscopy program or something along those lines. So you need to consider what the data are, what it is supposed to look like, and how you will be able to open it. Another way to do this in sort of a more secure way, something called write blocker, which connects to the hard drive or the external drive that you have acquired and allows you to open those same files on your computer, but it basically blocks you from making any really stupid mistakes like deleting something or saving over a file, which if you have automatic save on is potentially a problem. You really want to maintain the information that is associated with those files in their original way. So again, we've got a couple of programs. Some of these are free, some of them cost a lot of money, and some of them are more user-friendly than others. But this allows us to look at the metadata contained within a file in particular. So when we talk about metadata, we're talking about things that are internal to a program and things are possibly external to the computer system. So internal metadata includes when files were created, who is the author of that data, who was the last person that saved it, and what is the file type, just basic information. And that can usually be found in file properties or information in Microsoft Office. So this is my PowerPoint presentation. I seem to have not copied the place that actually says my name and the date, but it's located within information. This is within Adobe. If you go to the menu and go to document properties, it will show you this basic information. More external metadata might include the equipment that was used. So if you are taking an image of tissue or cells, it can include the information about the microscope used or the parameters of that sample collection. And those are important information to have for both investigations and for publication of the science. So what some of these programs look like, this is, these are just examples, so it's not important to necessarily be able to see what is on the screen here, but this is an image file. So images, your hard drive, and then basically program files, system volume. It breaks it down into ways that you can look at the data. So if it's images, documents, etc., it will break down the entire hard drive or a person's computer into one place. So Autopsy is a useful program. Arsenal Image Mounter, again, you're mounting an image and making it accessible for searching. And then Celebrite is Blacklight's new name. This is also a pretty great user-friendly program to have that image file or that EO1 file open so that you can search through and sort of index anything that you might be looking for. So what is that? What do you do with all that information? Once we find something that we're looking for related to an allegation, we might need to use plagiarism detection tools. We might need to use image analysis tools, and we might need to go crazy with some AI tools. So starting with plagiarism detection, there are hundreds of software out there for people to use, some of which are better than others. And this is, there are 3 slides here about these programs. This was put together by Stefan and a woman named Catherine Stebbins at Harvard that work with our Association for Research Integrity Officers, has a website, and the forensics group that we're a part of has all of this wonderful resource and information about these types of programs. So I'm not going to focus too much on these ones because they're all actually pretty terrible. And some of them aren't even free, which is sort of wild. So this is just sort of showing you that there are a bunch of different technology tools out there to use to detect when text has been copied. This is where sort of like the middle ground, when people are more comfortable using things like Google Scholar, and Adobe Acrobat. And these are mostly free, with the exception of Adobe making people pay for their license yearly at this point. But they're familiar, so it makes it a little bit easier to use. It's a good place to start if you're trying to detect some sort of text duplication from somewhere. But you're limited in your comparisons. So you have to sort of already have an idea of maybe what things should be searching. So with Google Scholar, it's at least searching within its own sort of database, but this is still pretty limited. And then importantly, the security of your documents and putting it sort of out there on the internet for searching is something to consider. So the tools that we recommend and things that lots of people use at this point. So Authenticate is one of the major programs. We do pay for a license up to a certain number of documents that you can put through here. Some of these are free, but they are known to have good security. They produce detailed reports and good results. But it does take a little bit of time to process sometimes. And so this is an example of what Authenticate looks like. And it's something that's important to point out here. We call this plagiarism detection software. It's not. What it's detecting is text duplication. Here shown in red, this is an example again that Catherine Stebbins did for our ARIO group. What is red, text in red here, matches with an article found on Nature. And then it just goes basically lists the percentage of text that overlaps with a particular source, and you can go in and look at what those matches are. There are different kinds of plagiarism where you're just taking an entire chunk and copying that text, or you're interspersing it throughout text that you have added to try to make it less match. But something that's important to really think about is looking at that percentage of text that matches or overlaps doesn't necessarily mean that something is copied in the way that you think it is. So take that percentage, but also make sure that you're looking and understanding what the output of these programs are. So again, text duplication can be assigned a metric, but in order to determine that something is plagiarism, you need to speak with people and have an idea of what the context is here. So what other things are we using then? So plagiarism aside, we're talking about image manipulation in the office. And again, we're sort of looking at the Office of Research Integrity at the federal level in the United States. Has provided some really wonderful tools to be used within Photoshop. So Photoshop is sort of not the gold standard, but it is the primary tool that is acceptable to use in research integrity offices in the United States and probably elsewhere. And what the ORI provides, and this is free to anybody that goes to their website, are things called forensic droplets. And these primary tools are things called gradient maps, which already exist in Photoshop itself, but the ORI has sort of produced their own, which may or may not help with is sort of identifying these contrasts and artifactual differences or similarities between images. So Stefan had some examples of what the gradient map looks like, and we'll look at it in a second. And then also this idea of an overlay. So if you take two images that you suspect are identical or highly similar, you can overlay them in sort of this way that shows what overlaps in red with a uniform border. And then everything else that is unique to one image or the other appears in different colors. And so what's important to note about these programs is that whatever, there's a quote, whatever inconsistency is revealed by the application is only an indication of manipulation or tampering with the image. So it is not a finding of falsification and not a finding of research misconduct until you go through the process that we've talked about. So having a look in Photoshop, your basic abilities, you can adjust brightness and contrast without having to know much about software that you're using. That's just going to an image that you've loaded, going to adjustments. You can do brightness and contrast and mess with the levels there. You can go to something called invert, which inverts the color in the black and white scale that might allow you to see some additional sort of artifacts around what you're interested in. And then gradient map is further down on this list here. And I just have a picture sort of what the gradient maps look like. The useful gradients A is what the ORI you can upload to your Adobe program, but Adobe does come with its own gradient maps as well to use for image analysis. So what does a gradient map look like? So this is an example of something that came across our desk, and here this band looks highly similar to this band with the exception that they're going in opposite directions. And so when you do, there are two different examples of gradient maps. You can see indications of splice lines throughout the image, but you also start to see more artifacts behind these bands themselves, and just a very sort of similar background patterns, etc. Just sort of gives you something to start off with. So when we look at that in a more sort of close way, so this is your band going one direction, your band going the other direction. If you flip it, you can start to see that these are even more similar than what they look like when they are vertically swapped. And then you can start to pick out artifacts, and we go sort of by the rule of too similar to be different. And so you start to look at artifacts that pop out in a gradient map and areas that are highly similar from one part to another. You start to look at the background, which would have been clear in a black and white sort of image, or at least very light. But you start to see just these entire areas of the image that are way too similar between these two things to actually be different. Another way to show too similar to be different is the idea of the overlay, which I mentioned as a tool. So here's your forward-facing band, your backwards-facing band, and we flip it, and then we can overlap them. And so this is a pretty great overlap here, solid red with no sort of fuzzy white or black background around it, and the other bands that are a part of that picture. And so we can come to the conclusion that those bands are the same, but then we need to continue on with the investigation to determine, did someone do that by accident? Did somebody do that on purpose? Can we find the raw data for these bands, etc.? So those are, the primary image analysis things that we do are related to Western blots and tissue images, and we do most of our forensics within Photoshop's program. There are additional analysis tools. Again, PowerPoint, I'm assuming that everybody has access to Microsoft's programs, so it's always a good place to start. You can do your basic brightness and contrast manipulations in Photoshop itself when you import a figure. It's not ideal, but it can start to give you an idea of if something seems off about an image. ImageJ is also a free program from the NIH. Anybody can download this program, but its user interface is not very user-friendly. So you need to have an idea of commands and codes and things within the program. If you are a scientist that has worked with microscopy images before, that's a benefit to using ImageJ. You probably then know at that point how to use this program, but it is not as friendly as it needs to be, but at least it's free and it exists. Forensically is a website that you can upload, I believe, JPEG and TIFF images to, and it has some very basic forensic analysis, such as like a cloning tool. So it will detect when somebody has maybe taken an image, part of an image, and cloned it and stamped it somewhere else on the image. It is not very reliable, but it is still a place to begin if something is suspected. And then the sort of idea of AI tools that have been emerging, I'm probably not going to talk about those nearly as much as people want me to, but when we think about AI tools to detect some sort of research manipulation, you have these machine learning programs that are predictive, that train on algorithms of historical data. So you have a data set. The examples for forensic software are ImageTwin and ProofFig, which I will go over. And essentially, these programs have been trained on sets of data from either PubMed or images that get uploaded to the program itself, or a database that they have to train the algorithms on. And then there's generative AI, which is your ChatGPT, and your Photoshop itself has programs for AI, where this can actually create new content. So you can ask it to please make me a Western blot, or please make me a sample of tissue to represent the liver, etc. But additionally, we can ask it to find those types of information as well. Can it identify the information that has been manipulated? So we get into sort of messy place. But the two primary programs that are used, you have to pay for subscriptions, but you can also ask these companies for trials, if it give you access for a few months or a certain number of publications or images to upload to allow you to get comfortable with the software. So ImageTwin does plagiarism detection, so it is incorporated into its program. You do not have to have a separate program for that. And then it does image manipulation detection. So it's comparing images within a publication that you submit. So if you upload an entire PDF file of a publication, it will compare the images within that paper, but it will also compare against a database. They claim of over 50 million images. And something important to note is they do maintain the privacy of the data that is inputted. So even if I can't recall actually if it's training on the images that you load, I feel like no is the answer. They will not like save that information as a part of the AI's learning process. So that's, you always want to be careful with what it is that you're inputting into any of these programs. So an example of what its interface looks like then, you have uploaded this entire PDF file. It's going to outline and identify all of the images, and then it's going to identify places where something might be duplicated. In this case, you've got a bunch of images of cells with portions of images that seem to, oops, oh no, cancel, snooze. Okay. Click cancel to restart. I don't want to do that. Okay. Sorry. So you've got portions of these images that overlap. They will sort of, again, output this number of how sure it thinks it is that something has been copied or not. And again, do this with the idea that this is sort of a screening process or something to start with. And I'll talk about why that's important in a moment. So the other example, the other software is Proofig. Again, this also does plagiarism detection, your image manipulation detection, and it does claim to be able to identify AI-generated images, although I have not used that myself to determine how well that is working at this point. It compares images within a publication, just like ImageTwin, and it actually compares against the PubMed database. So it has this large source of information to make comparisons to. If you have inputted a PDF file of interest, it might identify similarities in a different publication by a different person. So that data perhaps originated from a paper that had been previously published that somebody has manipulated and republished as their own data. So that's a nice way to compare things, to know where things are coming from, because PubMed is essentially our source of most scientific information and publications. And then something else that Proofig does do, it provides its own forensic tools. So similar to the Photoshop gradient maps and different ways of looking at similarities in bands and tissue, it has its own forensic tools within the application. So a lot of journals and institutions have partnered with ImageTwin or Proofig in an attempt to sort of scan through papers coming out from those institutions, or the journals are scanning things before stuff gets published and things like that. This is Proofig's interface. So uploading, validating, reviewing what it's found, and then it gives you a nice report. So does ImageTwin. But the types of manipulations that they're looking at, this is flipped, this is cropped and rotated, rotated, flipped, looking at splicing, looking at overlap of portions of figures, and then this here they claim is an AI generated image. So these are good for lots of reasons. The AI tools that we have, important to note then. So we're using these applications as technology tools. but they are not a determination of research misconduct. So if these programs output that something is identical, you need to follow up as the Research Integrity Office with your own assessment and your own forensic analysis. So you can put these images into these programs, but do not rely upon them as being the final thing that gets submitted to any sort of oversight committee. So it is really important that these not replace the forensic analysis that are done by our offices, but that they supplement things if necessary. So again, they can't determine if something is research misconduct. They can only identify a manipulation. And AI itself cannot determine the intent behind something. It doesn't have the whole story. It doesn't have the information that we can acquire from speaking to people in interviews and looking at the actual, source data itself if we can find it. AI tools can also be used then to generate text, as we know from the fact that this is already used in publications and in students' work and things like that. But it can also potentially be used to generate fake data. And that type of modification may or may not be identifiable in the ways that we know how. So Proofig is claiming it can identify when something has been AI generated or not. But this is an example here from Blake Talbot, who is sort of in charge of the forensic group of ARIO. And this is an example of putting in heart tissue to DALL-E with no reference image. So you're not providing an image of what heart tissue scientifically looks like. You're asked to be in terms of being able to make complex data and for your paper to falsify. You can't just go and say, please make me a Western blot. It's going to take what you input into it is really just important about AI. And at this point, institutions need to have policies in place for what is an acceptable use of AI in both text and images that are being published from institutions. And with that, I just made my cutoff time, a 2-minute clock. But that is my presentation. Stefan and I are available for questions at any point. And thank you so much for listening.